Okay, let's get started. Good morning, everyone. My name is Scott Teed. I'm a senior scientist and ecotoxicologist at Stone Environmental. I'd like to also introduce, he's not visible, but Sebastian Castro, who's the co-chair of the webinar series here at Stone. Just a brief introduction to Stone itself. Many of you on the call I know us, but for those who don't, we are an employee-owned environmental consulting firm headquartered in Vermont, but with offices in Canada, Austria, and Germany. Our vision is to lead development of innovative solutions that promote environmental sustainability. And our mission is we provide scientific tools, information, and analyses to help our clients solve environmental challenges with integrity, expertise, and innovation. And I hope many of you on this call have experienced that. We are dedicated to a collegial work environment that fosters excellence, creativity, and accountability through employee ownership. So this is our second year of our webinar series. It's organized by the Environmental Systems Modeling Group at Stone Environmental. This is the first event for this year, but we'll have more webinars in 2025 with topics to be determined. And in fact, after the webinar, you should receive a short follow-up survey. And one of the questions will be regarding which topics you'd like to hear about in the next 2 or 3 webinars. So if you can take 30 seconds out of your time and complete that, send it back to us, that would be very helpful. So in terms of webinar dynamics, we are going to turn off and have turned off all the mics of the participants and the video during the webinar. If you have questions during the presentation, please use the Teams Q&A feature at the top of your screen. Q&A will be at the end. We will restore your ability to ask questions directly. And if you would like more information on today's topic or have any questions about our expertise in other areas, we would love to hear from you. Please feel free to contact us through our website or LinkedIn pages or directly using the provided email address. The webinar is being recorded and all materials, including scripts, will be available for access, and we will provide a link in the chat box to access those at the end. And for those of you who know me directly, if you don't get that link or there's an issue, that just let me know and I'll provide it. So with no further delay, let's introduce the talk and our presenter. The talk today is on probabilistic tools for addressing uncertainty in species locations and pesticide use. Jonnie Dunne is going to provide us with this webinar. He is an environmental modeler and geospatial scientist, and he's been with Stone since 2017. Jonnie was born and raised in the Pacific Northwest and is now located in Vermont and has extensive experience in environmental modeling and agricultural science. His recent research highlights at Stone include developing software to model the spatial distribution of threatened and endangered species, pesticide usage, and co-occurrence between the two, which we'll hear about today, and developing models to simulate 3D airflow and spray drift interception in plant canopies, which, as many of you can imagine, would be useful for NTA analysis, for example, non-target arthropods. Without further ado, let's begin the webinar, and here's Jonnie.
Thank you, Scott, and thank you all for joining us this morning. As Scott said, today I'll be presenting on probabilistic tools for addressing uncertainty in Endangered Species Act assessments. So my presentation today is centered on the Automated Probabilistic Co-Occurrence Assessment Tool, or APCOAT is the acronym that you'll hear many times through this presentation. So the key functions included in APCOAT are to rapidly produce probabilistic maps of pesticide usage, endangered species habitat quality, and co-occurrence between pesticide usage and habitats. This software, user guide, and case studies are all available to download for free at stone-env.com/APCOAT. In this presentation today, I'll be returning to each of these functions from multiple angles, and we'll have plenty of time for the questions at the end for each if I don't cover something that you're curious about. Finally, before I get this all started, I'd like to thank our colleagues at Syngenta for their support in developing this software and conducting associated research projects over the past few years. For the first part of today's presentation, I'll be walking through an introduction to APCOAT's main functions, then diving into the details of the inputs and results of APCOAT assessments. and how the results of each of the main functions can be used to advance endangered species assessments. In the second part of the presentation, we'll look at some examples of APCOAT results, of how APCOAT results can be used in several types of endangered species assessments, including analysis of pesticide use limitation areas, incorporating usage data into co-occurrence assessments, and improving aquatic exposure modeling assessments. So we began developing APCOAT in response to the fact that co-occurrence analyses for ESA can present several challenges. Among the challenges is the sheer number of analyses that are required when there are 1,600 plus species listed by the services and over 1,000 products registered by EPA. Also, the methods required for these analyses and the data that is available has changed over the years, but the more advanced of the possible methods do require greater effort. The fact remains, though, that co-occurrence studies require the best available data for potential use site locations, pesticide usage, and spatial distribution of the species. While deterministic methods have historically been used, it has also been recognized that all potentially pesticide use sites and potential species presence locations are weighted equally, and that the available tools using these methods do not account for downstream flow of pesticides and residues. On the other hand, in response, probabilistic methods have been recommended by the National Research Council in order to better characterize variability and uncertainty in potential use sites and species distributions. So, our overarching purpose in developing APCOAT is to improve the accuracy and efficiency of exposure likelihood characterization. And our objective when designing APCOAT is to create a tool capable of delivering rapid assessment of spatial co-occurrence between pesticide use sites and endangered species using the best available data and probabilistic methods. In the example figure to the right, which is an output from APCOAT, we have a preview of the types of map results that can come from the APCOAT reports. In this case, we're showing the co-occurrence probabilities for the Colorado pike minnow and atrazine applications on corn. Just a quick note, this map is showing the single maximum co-occurrence values across all lakes and ponds within each watershed here. So while at initial glance it may look alarming, there's a lot of red, the overall co-occurrence probability across all lakes and ponds in this region for this pike minnow is only 2.2%. But this type of map helps us to quickly identify which locations may be of highest priority for conservation. So again, this region and this region right here may only have one single pond or lake that's in this higher co-occurrence bucket, but across all lakes and ponds for this species with atrazine, we found, again, the co-occurrence is only 2.2%. So backing up a couple of steps, APCOAT analyses typically begin with mapping the probability of pesticide usage. Packaged with APCOAT is a state-level pesticide usage data set covering 315 pesticides on 6 commonly grown crops. Those are alfalfa, corn, cotton, rice, soy, and wheat, which cover 90% of ag production in the US. To produce a map like the one below showing the probability of atrazine applications on corn, percent crop treated statistics for each state are applied to the pre-prepared crop site probability maps, which is why you see some stark differences across the state lines. They simply don't use as much atrazine in Iowa as they do in Missouri, which is not reflected in deterministic methods where all sites are treated equally. The other half of the APCOAT probabilistic co-occurrence assessment is a habitat suitability map generated from species locations and observed preferences for an array of environmental variables that are packaged with APCOAT. The probabilities you are seeing here, with the warmer colors being the higher probabilities, are for the Carner blue butterfly, and they reflect its preferences for 6 different bioclimatic variables, including the average temperature during the wettest quarter and the probability that each pixel will provide suitable habitat. Note that here the minimum value used for habitat quality probability is 20%, which is a common practice in habitat modeling to remove areas that are not likely relevant to our interests. Once both of the maps have been prepared in APCOAT, the probabilities for each location are multiplied by each other, summarized within a zonal map and a report document showing, in this case, that the average co-occurrence is 1.5% across the entire range for that Carner blue butterfly and atrazine applications on corn. With one watershed in here showing the highest maximum average co-occurrence of 8.2%. And again, these are intended to help users quickly identify areas that might be prioritized for conservation or restoration. So now we're going to dive further into the processing behind each of the previous slides, starting with that pesticide usage map. As I mentioned before, there are 6 probabilistic crop footprints packaged with APCOAT, and these are based on 6 years of cropland data layer rasters per crop. And then each raster probability, pixel-based probability, is then refined to agree with the cropland data layer accuracy metadata and NAS agricultural survey acreage. So you can think of, for a given field, the process begins with how many out of 6 years was it cropped in any one of these crops, and then based on, is the crop mapping data high or low accuracy in this state, it's adjusted upward or downward, and then does the total crop acreage agree well with the NAS agricultural survey acreage, and then those probabilities are again adjusted upward or downward to better agree with each of these 2 data sets. Then we also have the pesticide usage database, which is used to calculate the state level percent crop treated figures using those compiled USGS ePest estimates for 350 pesticides, and then acreage for each state and crop from the cropland data layer. Alternatively, all users may also provide their own usage data, which can produce PCT statistics for counties or crop reporting districts if you have that higher resolution than the state level data that's packaged with APCOAT. The other option you have is that users can assume that all sites are treated for additional conservatism, essentially assume, again, put in the number of 100% PCT for all states to kind of go back a little bit more towards the conservatism that you'd find in a deterministic analysis. So here we'll zoom in on the pesticides usage map and discuss some of the ins and outs of the method. I chose the intersection between Missouri, Iowa, and Illinois to better illustrate the differences in PCT we saw earlier. So you can also start to think about, that you can even spot there are some differences between states in kind of cropping probabilities here. You can see the fields look a little bit different, and those are better reflected in the next few slides. But first kind of figure we'll get into is the percent crop treated number. So as I mentioned earlier, more atrazine is used in Missouri than in Iowa, and those are the PCT numbers you see here. So PCT, percent crop treated, is calculated for 6 years for each state, beginning with the calculation of the acres treated, which is the pesticide mass applied divided by an application rate specified by the user. Those acres treated are then divided by the total treatable acres for that state and year. So now you have a time series of 6 years of these percent crop treated numbers for each state, and then we select a statistic from that 6-year time series to use in any of the analyses. And here we're looking at the maximum over that 6-year range for each of the states. Again, to kind of conform to the expectation of conservatism that we've seen over the years in co-occurrence analyses, you tend to use those higher end of the statistics on the time series. So then those PCT numbers are then multiplied by the probabilistic corn footprint to produce the map showing the probability that atrazine is applied to corn in each 30-meter pixel. So here we see some significant variation in probability within and between states that we wouldn't otherwise see in a deterministic map, which would essentially just show one shade of green. So here we're seeing that the darker shades of green are higher probabilities of atrazine being applied to corn than the lighter shades over here. So you can maybe assume that there are a good number of fields between these 3 states that are your typical corn-soy rotation, where you would expect the probability of corn being planted in each year to be about 50%. But since we have higher rates of atrazine usage over here in Missouri, those 50% corn-soy fields are rated higher than you see them over in Illinois or Iowa, where atrazine use is much less common. So next we'll move on to the species habitat models. And so I'll start by mentioning that these are generated iteratively, and I'll get to why in a moment. But for now, know that each iteration models the observed patterns between species locations and multiple candidate environmental data sets using maxent. Note also that multiple fitting functions are used when modeling species preferences, including linear, quadratic, and discrete step regressions. So in the figure to the right, we see the input data used for these regressions. In the example species is the Atwater's prairie chicken, so it shows a high preference in these observation data for higher precipitation during warm months. That's this bioclim 18 as one of a series of pretty standard bioclimatic variables that have been used extensively in species habitat modeling. And it's also showing a preference for lower solar radiation in April relative to the landscape background. So again, how these graphs are produced is that within the entire range of where you find your species location data, within that extent, you randomly sample what the environment is, and so that's these background data points. And then you also sample those values where the species has been observed. So that's the observation points. And you can see that, by chance, randomly, you would expect this graph to look, the observations to be the same as the background. But since the species is showing some pretty clear preferences for, again, higher precipitation during warm months, lower solar radiation during April, you see some pretty clear trends that are then incorporated into the species habitat model. So backing up though 1 second, as I just mentioned, APCOAT iterates through drafts of the SHMs before arriving at a final model that balances model fitting and model economy. So in each draft, contribution of each of the potential explanatory variables is ranked and low-ranking variables are removed. So here the process begins with iteration 1. You can see that it's very high model fitness, but the truth is that there are tons of variables going into this one. The very first iteration begins with all of the possible variables. So I believe in this case, we had 35 predictor variables. The next step is a big drop down to iteration 2. What happens there is that we first assess the correlation between variables, and if there's high correlation between any two variables, the one that shows the higher contribution to model fitness is retained, the lower one is dropped out, and then all the other variables that are lower than a certain specified, user-specified contribution threshold are also dropped out. So this model is much simpler than, iteration 2 is much simpler than iteration 1, but that parsimony is something that we're looking for a good, realistic, usable model. So we continue through this process and through each iteration, we continue to drop out predictor variables that are not contributing highly to the model until we see a distinct drop in model fitness. So iteration 6 represents that final drop. So we return back to iteration 5, knowing that this is the highest model fitness we can get with the fewest number of variables. Again, trying to balance model fitness and model economy. So the final iteration that is selected will show the probability that a site provides suitable habitat given multiple site characteristics. In our Atwater's prairie chicken example, these characteristics are, in order of contribution to the model, you can see here, solar radiation during April, land cover preference, and then bioclim 18, which is the precipitation during the warmest 3 months of the year. I should also mention that the fitness of these models is evaluated using reserved location data at each step, and these diagnostics are included in the final report. So we have 80% of your location data is being used to train the data, and then 20% is being held independent to test how fit the model is and how well each of these variables are contributing to that fitness. So I'll dive a little bit more back up one time again into the species habitat model inputs. These are the predictor variables I've been talking about. We'll start with those, which are all packaged with APCOAT. So these include 35 terrestrial rasters at 800-meter resolution, relating to several different types of potential predictor variables, and then sort of similar themes in the aquatic variables. And we have two different data sets for the aquatic variables, one relating to stream and rivers, flowing aquatic habitats, and then one relating to pond and lake variables or static habitats. So the other half of the SHM inputs are the species location data, which must be collected by the user. I will say that in the APCOAT installation that's available for download, we have one example species that's included with that, and that species data was collected from GBIF, but there are several other sources of freely available species locations on the web. But I also mentioned that users should be careful about combing through the metadata to make sure that the data is suited to SHM mapping. We do go through this in the user guide, but the two biggest factors to look out for are the date of the observation and the spatial accuracy. So these data often include older museum records that may not be relevant to the current landscape. They also have records that have typically been obscured to protect sensitive species, which is partially why the environmental data packaged with APCOAT is of lower resolution. So you can imagine that it'd be kind of dangerous to publicly put out maybe the location of, say, a bald eagle's nest. So instead, that data is often obscured to the nearest, maybe 50 square meters or 100 square meters where you might find it. So that's why we have these lower resolution terrestrial rasters and lower resolution aquatic variables relating to streams or lakes that are larger features, so that the accuracy of the species location data and the accuracy of the site characteristic data are well matched. If you happen to find or are able to acquire very high resolution data from individual state natural heritage programs, for example, APCOAT does allow you to provide your own higher resolution data to do higher resolution species habitat maps if you'd like. So once both the usage footprints and SHMs are prepared, co-occurrence analyses may be generated. As I mentioned before, the SHMs are typically filtered first to remove areas of very low quality habitat. So here we have at least 20% for the rusty patch bumblebee example. So for terrestrial analyses, the SHM rasters are then overlaid with and multiplied by the pesticide usage probability raster, and the resulting co-occurrence rasters are then averaged by zone for mapping purposes and over the entire SHM extent for the species summary. In our example for the rusty patch bumblebee, we can see that there's a belt of higher co-occurrence zones that could be targeted for conservation, but again, across the entire range, the average co-occurrence is only 1.76%. For aquatic analyses, the co-occurrence methods are considerably more complex. In this process, we distinguish between features that are hydrologically connected to neighboring watersheds and those that are disconnected from neighboring watersheds. To characterize the usage within each watershed, we also calculate both the maximum pesticide usage value for conservative estimates and mean pesticide usage value to better understand the relative exposure magnitude. So hydrologically connected features include those that receive flow from neighboring Huck 12 watersheds, such as these mainstream rivers right here. So you can see this is the water flowing from upstream to downstream. So we have a connected main stem feature right here, and the water bodies that are located on those main stem features. So you can see a couple of ponds right here. The disconnected features include tributaries, water bodies on those tributaries, and ponds, and so water bodies that are completely isolated with individually delineated pond sheds. Again, for each watershed shown here, we're summarizing the pesticide usage values and then multiplying those values by the SHM values for each hydrologic feature. So we go through that process in 4 different ways, again, incorporating both the upstream and just the local usage, and also the maximum and mean values in both those upstream and local usages. And those are, once you have those for each aquatic feature, those are multiplied by the habitat suitability values that are associated with each feature. So also included in the co-occurrence output is an HTML report document containing the results of each input analysis. So the results of the co-occurrence analyses include the average co-occurrence over the entire species reign, zonal co-occurrence statistics, the zonal co-occurrence map, and again, multiple flow tracing methods used for each aquatic species. And those are all reported separately with the mean, max, local, and upstream usage being calculated. So to summarize quickly again, the results of each component, the pesticide usage results include statistics derived from PCT calculations, which account for variability in pest pressure and pesticide usage over multiple years and states. as well as high-resolution maps of pesticide usage in the US, which account for variability in cropping practices. The habitat quality modeling report includes summaries of the inputs, selected predictor variables, and model fitness statistics. The co-occurrence report includes a convenient summary of the species range-wide co-occurrence probability and a high-resolution map of co-occurrence for use in conservation and restoration practices. Looking back at the bigger picture, we have identified several ways that each of the APCOAT components can advance endangered species assessments. Broadly speaking, the fact that APCOAT recognizes uncertainty in all components of co-occurrence analyses means that it provides objective analyses to better understand species locations and usage restrictions. The pesticide usage modeling refines mapping through more comprehensive data inputs and processing. And these products could be used with existing range and critical habitat data to improve the realism of deterministic overlap analyses. The species habitat modeling leverages machine learning to maximize understanding of habitat requirements and provides objective analysis to better understand species locations and usage restrictions. And finally, the co-occurrence assessments provide automated reports to ensure transparency and consistency between analyses and make the best available data and probabilistic methods easily accessible and reproducible. So in these final few slides, we'll look at a couple example use cases of how APCOAT could be used to advance endangered species assessments. First, we can see how SHMs from APCOAT could be used to refine pesticide use limitation areas, or PULAs. So PULAs are to be based on the best available species location data, as well as potential pesticide transport into those locations. In the PULA development SOP, SHMs are identified as a potentially suitable core habitat map type for some species. So APCOAT is well positioned to contribute to those efforts. Additionally, SHMs could be used to identify potential habitat outside of recognized ranges. So here in our example of the Atlantic pigtoe mussel, we see that there are several high-quality habitats mapped in Virginia, for example, that could be targeted for monitoring for viable populations. So this example, though, also raises the important point that ideally these SHMs should be validated by other sources and, in a perfect world, field surveys. In this case, for example, Fish and Wildlife notes that there are 14 known populations in North Carolina, but only 2 in Virginia. So that may be why the U.S. Fish and Wildlife Service range data does not officially extend to cover these areas in Virginia. Fortunately, these methods are also pretty well settled science, and the APCOAT method does align with the Fish and Wildlife Service SOP for refining listed species ranges. So, like that SOP, Maxent is the base of the SHM mapping process, and the modeling process is very selective about which predictor variables are included in that final model. So the probabilistic usage methods packaged in APCOAT can also be used to refine deterministic assumptions and co-occurrence analyses. In this example, we extended the usage statistics to also simulate field scale applications of reported usage, which highlights areas where pesticides are likely to be used as opposed to where it is potentially used in deterministic analyses. So the results show that deterministic overlap analyses can be overly conservative, with the standard EPA deterministic methods consistently showing much higher overlap at multiple transport distances relative to even the most conservative usage statistics. So here again, we've got the deterministic EPA methods compared to a probabilistic co-occurrence method, where these are the different percent crop treated statistics that are used in a suite of co-occurrence analyses for both the range and critical habitat for a number of species. And we consistently see that where the deterministic methods, the number of species with high overlap is much greater with this method than with these probabilistic methods that we're talking about today. And finally, thanks to the high resolution flow tracing methods packaged with APCOAT, it can be used also to refine aquatic exposure assessments, particularly for characterization of upland pesticide usage for aquatic features. Conservatism in these analyses can also be adjusted through PCT statistic selection. As I've mentioned a few times before, you can go lower to higher statistics here, depending on how conservative you want or need these analyses to be. In the report that this example is drawn from, we also found that the differences in EECs, that's expected environmental concentrations, vary by crop and pesticide. But for this particular pesticide, which this figure is showing the results of, we see that the PCT refined EECs are consistently lower than the screening level deterministic method results. So looking ahead to the next phase of APCOAT development, we are excited to announce that we've just had a manuscript accepted for publication detailing our analysis of 375 aquatic species, available now in IEAM. This is, I think, the third paper in a series of research projects that we've done over the years with developing these probabilistic methods. So nice to see again that these are pretty well settled science that have been peer reviewed and accepted in the literature. Looking ahead to the rest of the year, our current development phase includes plans to expand the pesticide usage footprint functionality by increasing the number of additional crop footprints to cover all crops that are mapped in the United States, and allowing users to create custom crop group combinations for additional ePest estimate data groups. We're also expanding the offsite exposure modeling features to include custom drift distance probabilities and the additive probabilities of overlapping drift buffers. We're also figuring out how best to distribute the methods for conducting multiple realizations of field-based usage allocation. So that's the example that I had 2 slides ago, where pesticide masses are distributed across randomly selected ag lands to better mimic real-world applications. And finally, we are modifying APCOAT to conduct co-occurrence analyses using PULAs in place of SHMs, where the needs of a species may already be well understood. And again, to kind of look ahead to how those PULAs are going to be more and more incorporated into regulation of pesticides. It seems as the trend that we see in the coming years. So thank you again all for coming. Thank you to my colleagues, Hendrik Rathians and Mike Winchell of Stone Environmental, and Richard Brain, Max Beacon, Tony Bird of Syngenta Crop Protection, who have been, again, supportive of this project for several years now. So excited to take any questions that you might have. And as final, just in case we need the reference, a quick glance at what this software looks like and give you an idea, hopefully, of how easy it is to use. You can get a sense of, it just takes maybe a dozen clicks to get all of this all set up and running once you've got your input data prepared. So thank you again, and let's hear some questions.
I'll kick it off, Jonnie. Do you have any plans to deal with, to add non-ag data layers, for example, open space developed or something like that, which may be important, or mosquito use footprint?
I haven't thought explicitly about that yet, but now that you mention it, I do believe that that's something that we could be, could easily be done when we're doing these 90 additional crop footprints in this coming year. So, currently we're just covering those 6 crops, but this will now include all of the classifications that are available in the cropland data layer. So yes, open space, different degrees of, low, medium, high intensity development, those could all be included in APCOAT as well.
Perfect, thank you.
Yeah, the ePest data does not include those non-ag pesticide usage estimates. So you would also need the pesticide data to be on the user side, or as I've mentioned way back at the beginning, you could just assume that those are all 100% crop treated, or not crop, but 100% probability that they are treated. And now that I say that, I think that you could maybe even, no, it's just 100% crop treated, or 100% treated, but it would be easy, I think, to modify it so that we could do, any number across the board. You could say 150, 75% probability that any non-ag site is treated. And yeah, potentially could, right? Yeah, again, you need user specified kind of pesticide usage data to go all the way with that.
Okay, thanks.
Yep.
Are there any other questions?
Hi, it was a very nice presentation, and I can see how useful this tool can be. A commendable job. Just a question, was EPA involved in this development of this tool somehow, or was it more of like environmental sciences project?
Yeah, this was primarily Stone Environmental and Syngenta working together on this. But of course, we, we're very conscious of EPA's methods and sort of are drawing on, historically what they've done for the deterministic analyses. So this is essentially just refining those. And as I mentioned, that we're kind of conscious of their work on PULAs and are working to kind of get a little bit more feedback from them in this next development cycle to see how PULAs or how they might be able to use or modify this tool themselves.
Yeah, it's like a huge leap in this field.
Mhm.
So congratulations to you. And I might have missed it. How can we use this tool? Is it like available for downloading, or do we have to come back to you to use this tool? I might have missed it. Sorry.
No, it's fine. Happy to have another chance to plug it again, mention it again. Yes, so the software is available for free on our website. Pretty easy to just Google APCOAT or follow that link. I'm sure in our follow-up or in many spots, maybe somebody even in the chat could drop the link so you could go right to it. But yeah, the software is freely available. There's also a user guide and case studies that we did on the first version of the software where we did a bunch of analyses with atrazine and 10 endangered species. So we saw some of those examples here in the presentation. Yeah, so just to also give you an idea, this is version 2.2 of the software. So again, this is pretty continuous development of it. We hope to be able to keep doing that in the years to come to make sure that this is useful for all potential folks out there.
This is great. Thank you so much.
Thank you.
Jonnie, we had a question in the chat. How are wetland species handled in APCOAT?
Yeah, so we got a question about this on our most recent manuscript. So I'm glad to see that folks are thinking the same thing. So maybe I can pull that up, the aquatic methods again, just to illustrate this. So wetland species would probably be best represented by the static water bodies. So you got ponds and lakes data available that you can use for the habitat models, and what you would expect to find then is that you have a map of all of, say, your species locations for an endangered wetland species. Then you would anticipate that those species locations would be very close to any of these ponds or lakes, or even for the I guess for some types of wetlands, it might be appropriate to use the flowing data set. But backing up to the ponds and lakes, these are the environmental data that's associated with them does include a lot of land use data sort of within the first 100 meters buffering the area. So we expect that is a pretty good representation of the interface between the terrestrial and aquatic environments. And that, again, I didn't belabor the point a whole lot, but for the data sets that you find in the aquatic variables, they're pretty extensive. So we anticipate that of those 148, there are going to be some in there that are going to pretty well characterize that interface. And if you're interested, again, I'd recommend you go and find our manuscript just published, because we do talk about that a little bit more. And in the appendices, even for that, we do have the results of all 375 aquatic species. So you can yourself start to look at some of those wetland species that are involved there, and the broad strokes of the results that we got from that.
Thank you, Johnny.
Mhm.
Do we have any other questions? And while we're waiting, Sebastian has placed some links in the chat to access the APCOAT itself, as well as the webinar slides. So you can grab them there.
Go ahead. See the question from Brian. Is there capacity to model species dependencies, indirect effects, PPHD? Maybe Brian, if you can jump in. I'm not familiar with PPHD.
Prey, pollination, habitat, and dispersal.
Okay, thank you. Yeah, so yes, if you have that data available as a map, mapped data, then those you can add those pretty easily to the SHMs yourself. So you can see here, in the interface, if you're doing a species distribution model, species habitat model, you have the buttons to enter your species locations, and then you've got also the predictor variables that are packaged with APCOAT. But you can also point to your own predictor variable folder. So any type of data that you think is relevant and you have it available, you could do that. That first half of the question, dependencies, indirect effects. My mind first went to cross product effects, and Maxent does include some of that in there in the evaluation of the different model fitting functions. But yeah, it's also important to note that the correlation between variables is evaluated at the outset, again, in that very first model iteration. So variables that are highly correlated are dropped, are winnowed down pretty quickly to go to these more reasonable models.
Okay. Are there any other questions? Mike, unmute yourself if
Yep. Can you hear me?
Yep, we can hear you now.
Yeah, just following up on that question about the species dependencies. I think what you could do, and Johnny alluded to this by mentioning that you can add in your own data layers, but if there was some other species that the one you were trying to model was dependent upon, and you had a spatial data layer that represented the locations or the range of that species, you could use that as an input to APCOAT as another potential predictor variable of the likelihood of the species of interest being present in certain locations. So I think that could be done.
Thank you. Yeah, right. Yeah. So example, if you had a bumblebee that you knew was dependent on a particular flower, you could potentially, map the suitability of the habitat suitability of that flower, and then use that output as an input into the habitat suitability map for the bumblebee. It's another question we've gotten from reviewers of manuscripts before. So.
Okay. Any other questions?
You've got my email if other questions come up to you later.
Yeah, absolutely. Please feel free to contact us at any time to discuss this. I'd like to thank Johnny for an interesting, informative webinar. Thank you again for your interest in the webinar series and for Johnny to start things off in 2025. We will, as I mentioned before, send you a short survey. So if you could take 30 seconds and just return it to us, it would be much appreciated. We're looking for recommended future topics for webinars and to help us improve the webinar experience. So stay tuned for more information on upcoming webinars in 2025. And if there's no more questions, then have a great day.
Thank
you very much.